Go unfortunately very deep and low, okay? So do go as much as you can, just make sure you're not hitting the others. We're gonna do it first, slowly, okay? And talk about what we have, and then we do one quick round, okay? What you are not allowed is to move up until I say so, okay? So we start here, we're focusing on the quadriceps, and you can put your hands so you can feel the quadriceps, okay? So we start, okay, so we start like this. Let's think about the quadriceps. We are now going to go down, okay? So let's just go down. Again, I can only do this, but Diego, why are you standing? Okay. Now, gravity pulls you down, right? Your quadriceps are resisting. Now, if you just staying still, I did not say go up. I don't know why you're doing. If you just stay still, what is the type of contraction? Isometric, right? No movement. But at some point, right, it's starting to pull you. Are your quadriceps working now? They're contracting. What type of contraction as they are lengthening and extending? Eccentric. Contraction, isotonic, eccentric. At some point, do it slowly now. We're going to gather enough myosin heads binding to active filaments, and we're going to go to concentric now. We are shortening, okay? So now we're going to do one more round, but now we're going to say it slowly. We don't want to do it slowly, but now we're going to say it as a group, okay? So let's do, first of all, it's going to start also with right concentric as we continue. And now, iso, I don't hear you.
Metric.
Isometric. And now, as we're starting to pull down, quadriceps contracting is eccentric. We want to go up, so we start to pull and it's still isometric, but at some point we're going to go, what type of contraction?
Concentric.
Isotonic, concentric, you can see down, let's continue.
Yeah. That was so fun. Probably how I feel about that time. Yeah.
Quadriceps are working differently. Well, as you go down, it's not the ones that may be actually pulling you down so much. The quadriceps start to work mainly when they go up. I do want to say one word. We're not going to analyze this, but if I had asked you about let's go back to the molecular structure, the sarcomere, what's going on here, you will not be able to answer me. What exactly happens at the sarcomere level? This is one of the, we talked about the sliding filaments theory, and we explained why we call it a theory, right? This is actually one of the weaknesses of the sliding filaments theory, because it's very hard to use the sliding filaments theory to explain what's going on here. There are some nice studies that probably show the strong evidence that we have right now is that the elastic fibers are having a very important function here. I'm talking about, eccentric contraction, just to make sure we understand. Okay? But just to note, without going into the details, that a theory, even when it's established in good theory, does not necessarily cover all small questions, and this is one of them. But we're still struggling to make sure we understand at the molecular level. Okay, so we're continuing with physiology, and we're going to talk about muscle responses, how the muscle responds to the action potential. It's contraction, of course, but what happens during that time? So to do that, we first need to introduce a very, very important term, because we talk about a muscle organ, and it's called the motor unit. In this example, you can see a muscle organ, again the biceps, it's always the biceps in those diagrams. And the muscle organ contains multiple muscle fibers, right? Elongated, running along the muscle organ, the length of the muscle organ. In this case, we don't have a lot. Of course, we have many more in reality. But we also label them in different colors. And if you go back to the spinal cord and the somatic motor neuron, remember these are somatic, right? Because we're talking about skeletal muscles. What we can see is that one neuron, let's look at the purple one. Cell body is purple, the axon is purple, and here you have the axon terminal branching. So what you'll have, that one neuron is going to innervate, reaching out and control several different muscle fibers in that specific organ. In this case, all the purple labeled muscle fibers are innervated and controlled by the purple neuron. Another neuron, we have 3 here, will innervate the red muscles, and another one will be the blue. Again, all of them are part of a muscle organ, a given muscle organ. All of them will go together as part of a huge nerve that is comprised of many, many, many neurons. But they're split at the end to that part. Motor unit is composed of that somatic motor neuron and all the fibers that that motor neuron innervates. So a motor unit is defined as a somatic motor neuron and all the skeletal muscle fibers it innervates. How many motor units do I have here? 3, right? So the purple motor unit is the purple neuron, labeled neuron, and the purple muscle fibers. And as we will see, when we have many muscle fibers in a muscle, since all of the muscle fibers that are shown in purple will get the same action potential. Once the purple neuron fires, they're all going to behave and respond the same, right? Now, you'll have one neuron innervate multiple muscle fibers, but if you are a muscle fiber, you will see only one neuron. Okay, and that's very important. Otherwise, you will have just one big mess. You have a question?
Yeah. Is it intentional that the different muscle fibers for each motor unit are about evenly distributed throughout that muscle then?
No, it's not going to be. It's not completely random. Okay, so you'll have a nice distribution, but it can be that one motor unit will be composed of, let's say, 15, and the other one will be with 50. It doesn't have to be exactly equal numbers, but they will be spread out throughout the muscle or distributed along the muscle. Now, everything that we will discuss when it comes to how a muscle responds to an action potential is going to be discussed at the level of the motor unit. Because it might be that one motor unit will fire an action potential, while the other one will not. Once the blue neuron is going to fire an action potential, will all the blue muscle fibers respond? The answer is yes, because they are all getting the action potential. The reds, they don't know about that, right? They need to get action potential from the red neuron. The second term we're going to talk about is what we call the muscle twitch. The muscle twitch is the response, and when I say response, I mean the mechanical tension of a motor unit, not of a muscle organ and not of a muscle cell, but of a motor unit to a single action potential. Basically, the purple neuron just fired an action potential. What will be the response of all together, of all the purple muscle fibers? Not one of them, all of them together. Any questions about the terminology, about the definitions before we continue? Okay, so this is a classic, typical twitch. On the x-axis, we look at the time in milliseconds. On the y-axis, we look at tension. This is not tension in kilograms. This is tension as a percentage of the maximum tension that this specific motor unit can reach. And it can be different between different motor units. Okay? But we are talking about a max. We're starting, of course, with 0 tension because there's no pulling here. There's no pulling here. And now we divide it into 3 parts. The latent or latent period, the period of contraction as we develop more and more tension gradually, and then a period of relaxation. Let's start with this question. Anyone wants to address why do I have this latent period? It's very short, right? It's just a few milliseconds, something like 5 or 3. Why do I even have what makes that happen? Yeah. Part of it will be calcium leaving. What else needs to happen? Remember, we just generated an action potential. A stimulus here in the muscle, right? Came from the neuron, but now it's here. What else needs to happen? That was right. The release of calcium. Then we need an ATP to bind and to hydrolyze, right? What needs to happen with the action potential that just came and was generated in the neuromuscular junction? It needs to move to, right? So basically, we called all of these events, we gave it a term. Anyone remembers? It was almost 2 days ago. Excitation, contraction, and coupling, exactly. Excitation, contraction, and coupling is what's responsible for that little short window of time until we will start to see myosin heads pulling. Only when the first myosin head will start pulling, that's when you start develop tension. Okay. So then we have the muscle contraction, more and more and more myosin heads are starting to pull. And at some point they release. We talked a lot about relaxation, right? And this is the relaxation part. So 3 phases. Now, all motor units will have a muscle twitch, and the pattern will always be like that, but they can be different. For example, here are 3 examples. Now, before we even analyze them, I want to make sure we understand. You see different names of different muscles, gastrocnemius, the soleus, for example. But the muscle, this is not accurate, actually, because the gastrocnemius will have different types of motor units. The difference when we compare our muscle organs, the biceps compared to the gastrocnemius, compared to others, will be in the distribution of motor units inside the muscle. But the muscle typically would not have exactly the same motor units. So let's talk about this, and we'll come back to the whole idea of distribution. So this is the soleus, for example. Where's your soleus? Okay? Below your hamstrings, right now. Think about what the function is. As I'm standing, I'm not doing anything, my body joint, my top part of the body is sitting on my muscles. So my soleus needs to contract, right? And to make sure I'm not collapsing, basically. How do we call that part when even if I don't move, I have some of the muscle working, muscle fibers? Tone, muscle tone, right? So soleus needs to endure, if you will, heavy weight for a prolonged time. It's not so much about being fast, it's more about being stable and endured for a long time. And indeed, you can see that it takes some time to build the max tension in the soleus, but the whole twitch is pretty long. You reach full relaxation in 200 milliseconds. If you compare that to the lateral rectus has nothing to do with your rectum. Okay, lateral rectus, where is that? Eyes. This is when you move into the left and the right, the eyeball. Okay? And look at the twitch. Again, it's the same amplitude, just because, remember, it's not kilograms. It's percentage of the maximum potential tension of that given twitch. So you always have the same max. But look at it twitches its top, max, contraction in like 10 or so milliseconds, and it's done at 20 milliseconds. Why is that? Why do we gain from that? Why do we see a different twitch in there? Think about the function of the lateral rectus. Right? I don't need to carry a huge load. It's not my body trunk or anything like that. It's just an eyeball, right? But I need to be very fast. And I'm not just fast to contract, but also fast to relax, because I'm looking to the left, and immediately after that, I'm going to look to the right. So you can see function follows form, right? You can see how the anatomy dictates the physiology, and it has different roles. Gastrocnemius is somewhere in the middle. Where is the gastrocnemius? It's just beneath the soleus. Sorry, not beneath the soleus. It's surrounding the soleus, right? So it does not carry as much, but it's still not the lateral rectus. At the same time, we're going to use the gastrocnemius when we jump, for example. So you also need to be pretty quick compared to the soleus. Okay? So you have different responses. It's the same pattern, the same 3 phases, but different timings of each of these. And this is the muscle twitch. Okay, and we talked about this. I forgot to put this, the excitation, contraction, coupling. Any questions about the twitch? I'm going to challenge the twitch even more, but yeah. Is there? The composition, you mean, of motor units in a given muscle? The answer is yes, but with use, but it's not like from this minute to the next minute. So it's not as dynamic, but yes, it will change during usage, I guess. Okay. So I want to move that with the twitch and talk a bit about our muscle response, right? And who is this guy? Let's see how many first years and sophomores and juniors I have here. Juniors should know, right? Who is this? Kennedy Nix, right? And every few semesters I'm changing the picture, right, to a more, it used to be Tyler Hansbrough all the time, because that was a perfect example for this. We don't really have from last year like a natural center, right? And I thought, okay, we also have national championship, so it will be nice to remember. I'm showing you Kennedy mix because he was a center and he was very, very strong. You can even argue he was violent if you were forced the guy fighting against him, trying to get the rebound or trying to defend him, putting the dunk. You don't want to be this guy, right? Basically. At the same time, Kennedy used to have a very nice cat, a kitten, a small kitten that he loved and he still loves, right? And one of the things he loves to do is just to he graduated on his homework, but having a cup of tea and petting his kitten. For the sake of his kitten, right, we really hope that he's not confusing right now with a basketball game. These are the same muscles that Kennedy used when he pet his kitten and when he slam dunk, right? The same muscles. So we need to understand how we can use the same muscles and grade our response. How we can use the same muscle to make a weak contraction, if you will, versus a strong contraction with the same muscle. So, close your laptops because I want you to see the next slide. Okay, or print out, just hide them. Let's talk with each other and think about what mechanisms can potentially allow Kennedy to use his muscles without hurting his cat. Okay? How can we grade our responses? Before we start discussing this, a question for you. I'll just suggest an idea. Maybe we'll send stronger action potentials from the neuron. Can we? Once we'll just do +30, and the second time we'll just do the +50. No, right? Because you must right now, the nervous system, you know that an action potential will be an action potential, so it cannot be a different action potential. So think about two types of mechanisms by which we can do that. Let's talk with each other. The frequency, wonderful. We'll talk about it shortly in a second. Maybe we'll wait the second one. Okay, let's talk about frequency first. So increase the frequency. We talked a lot about this when we talk about the nervous system, is that one way we talked about EPSPs back then. In this case, it's a bit different, but one way is that if I have one neuron firing an action potential, I can fire it more rapidly, right? We will always be limited when we talk about frequency with what, or by what? With the absolute refractory period. We cannot do it every half millisecond, but once the absolute is done, we can fire more and more and more. So let's take a look at the twitch as we increase the frequency, and then some new terms. So this is the regular twitch. Latent period, it's hard to see, contraction, relaxation, again, S for stimulus, right? So I'm generating a stimulus from that, again, purple labeled neuron, then all the motor unit, all the purple muscle fibers will contract after a latent period, and then at some point, we're done, we rest, we wait a bit, fire again. So, question, Emma?
So is this kind of like summation?
In a way, it's like summation, but let's see the summation. You're right. Okay? Now, I will ask you a question first. So, now I fire it again after a stimulus, right? Now it goes contraction, contraction. Great, we reached the relaxation period. Now we fire in the middle. My first question to you is, what do you expect it to be? Are you going to go all the way down and then up, or are we going to stop it and now we continue up? Up or down first? Up. We'll see why in a second. When we reach the up, is it going to be the same level, or can it be even higher? It's going to be higher. So this is what we call wave summation, as Miranda suggested, right? Wave because it looks like a sine wave, okay? So we have two phenomena here. Phenomena, I guess. One of them was that by increasing the frequency, look at the frequency of the stimulus, right? Which is the action potential in this case. We did not reach complete relaxation, but not only that, we even reached a higher tension that we had. Who wants to suggest what's going on here at the level of the cell, of the cells? What's happening inside? Yes, Ira? Not here. We're talking, this is a twitch. Remember, a twitch is just one motor unit, right? You're talking about a muscle organ, we'll talk about that. But right now we are in the realm of one molar unit. So what's going on here? What is responsible for the going up and even higher than what we had? Think about what provides contraction. That's the hint, that's the key. If I count to 3, can you tell me a word? What is essential for contraction? 1 to 3? Calcium, you got it. Okay, so think calcium. What's happening here? Yes.
So when you have that second potential that comes through and has the summation, you get even more calcium released into the cytoplasm. So it kind of amplifies the already working mechanism.
Exactly. Wonderful. Imagine we are here. I'm in the cytoplasm right now, okay? And we have calcium, right? Because we stimulated and we had action potential. And now I have myosin heads binding to the actin filaments, right? Gradually, more and more myosin heads will bind to the actin filaments. What happens during relaxation? What needs to happen to the calcium? It needs to go where? Back to the terminal cistern, right? Now imagine, I don't know if you guys can all see that there's a door, there's an exit door right here. Imagine that this door is leading to the terminal cistern, right? So what we will have gradually, not all of them at once, are going to lead to the terminal cistern, right? But if I stimulate it again, the muscle, then now the doors open again and more calcium is coming in before the entire set of calcium ions were able to go back. I still have myosin heads bound to the actin filaments. Let's just throw numbers. Let's say I had 1,000 myosin heads bound to the actin filaments, right? And now I had some calcium going back, so now I relax. 1,000, 800, 600, but suddenly I have more. I'm not starting from scratch. So I'll have even more than 1,000. I'll have 1,200 myosin heads will bind to actin filaments because now I have more available calcium. I did not allow all the ions to go back to retrieve to the resting state. I flooded the cell with more calcium. What will happen if I do it again, but now even with increased frequencies? I will continue to go up. Okay, so new stimulus starts before the calcium is released back to the SR, to the sarcoplasmic reticulum. If I continue, I will continue with the wave summation. We call this phase an unfused tetanus. Tetanus, nothing to do with the tetanus with the bacteria. That's something to do with the nervous system. Okay? We just call it tetanic contractions when we are contracting one after the other. So it's going to go up and going to go up. Of course, there's a limit, right? Because I can just saturate all the myosin binding sites on the actin filaments. And if I continue to increase the frequency, look at the frequencies here, at some point I will reach something that is called a fused tetanus. I cannot contract this muscle more than I'm doing right now. Literally, all the calcium ions are there, everything is hooked and connected, and it's now just a cross-bridge cycle depending on the ATP. That's the idea. Typically in physiological conditions, when you just work out or do something, you will not reach a fused tetanus, but you can definitely, right? So that's Kennedy mix, right? Kitten, that's Kennedy mix in a game against Duke. That's a Duke part. Okay? Now, if you continue with that for too long, that will reach muscle fatigue. Unfortunately, I'm not going to talk about muscle fatigue, and the textbook does a pretty bad job, I must say, about that muscle fatigue, unfortunately. But at some point, we are not going to be able to continue, and we'll have to go to relaxation. Yes?
So, would you reach the maximum possible amount of muscle contraction before the fused tetanus?
You will reach the maximum contraction at the fused tetanus. But again, if you continue with that, because that's the max, right? Basically now, all the calcium opened up all the possible myosin binding sites of the actin filaments. And then the myosin pull, that's your max tension.
Is the magnitude of where we get fused tetanus change how fast you get there?
Is the magnitude? No, because you eventually what you have, the magnitude will not change after you reach, or how fast you reach, how quickly you reach, because it depends, it's all about the calcium and how many myosin heads can bind to the actin filaments. That's the idea. That's a fused tetanus, unfused tetanus, wave summation. So unfused tetanus is basically a sum of wave summations. That was the first mechanism, changing the frequency, right? The second way to change the strength of the stimulus and the response would be to do what? You guys covered this. I heard it from many. What are you going to do? This was one motor unit. You can change the frequency. What else can you do? I can just recruit more units, right? So if you think about my right arm, the arm to your left, right, as innovating, the motor unit is now Lucy and Crystal right here. You don't see them, you'll just need to believe me, right? So I am innovating Crystal and Lucy. Okay, these are two. Okay, and now I'm going to have Dana, Nicole, Emma, Sierra, four motor units, four muscle fibers with my left arm, right? That's my purple neuron, if you will. That's my blue neuron, if you will. So I can just decide, okay, game against Duke. What am I going to use? Both, right? So I have 6 muscles working right now. I'm resting, enjoying the game, enjoying the win. I'm going to pet my kitten. What am I going to do? I'm going to release this and just use one motor unit, probably the smaller motor unit, because I still need contraction, but not as much. So that's recruitment of motor units. And you can see that if you look at a nerve, and a nerve is composed of many neurons, right? And if you look at a cross-section of a nerve, each gray circle would be one neuron. So if you look at the cross-section of this, right? So one neuron that is stimulated. So here, none is stimulated, so the tension in this specific muscle organ is going to be 0, right? Now, if you stimulated 2, you have 2 motor units. It's not 2 muscle fibers. It might be 10, it might be 50. So you get some tension. You recruit more, you get more tension, you recruit more. At some point, you reach the maximum tension using basically all the motor units. So by regulating the number of motor units that are being stimulated, and remember, in each motor unit you have a motor neuron. So this is how we go back to the nervous system. Your brain right now tells you contract in the beginning of this semester, like first lecture, right? I told you about a circuit, and I told you about I'm jogging, I'm coming back, and there's a glass of water, and I'm sweating, right? And we talked about sensory and motor. And I told you that, okay, now I'm telling my motor, somatic neuron, to tell my muscles, hold the cup and contract my bicep. But it's not just holding the cup. I don't want to be too loose, right, because then it will drop. I don't want to be too tight, because then I will break it. That's how we grade that muscle response. So at some point we'll just say, okay, I just need to hold it, not too tight, so 2 units with this frequency, oh, it's about to drop, more units, increased frequency, all of this is coming from here. And I just want to briefly go and talk about, and just to, again, we talked about this, but just to remind you, muscle tone is a continuous contraction of the muscle, helps to maintain posture, okay? And this happens all the time, so we have some units that contract with some frequency. And I just briefly want to, kind of like a review of something you already read about, and that's going to be the energy, right? So we're going back to the question we asked, is that so much ATP is required, right? We talked about why it's so important for contraction, relaxation. Where do we get it from? Okay? So close your computers. We'll just do it for a minute. This is what we have for this. What I want you to do is together think about what we'll fill in the blank here. Okay? So she's about to run. Okay? She's starting to run, continues to run. You can see the duration, kind of like short duration exercise. What happens in the 10 seconds, in the 6 seconds, 10 seconds, 30 to 40 seconds, and then you continue for hours, right? She's going for a half marathon. So just think with yourself, and what would you see there? Think about it for a little bit, and then we have, first of all, ATP. We have some ATP in the muscles, right? We're going to use it. It's not going to happen for long. Don't stick to the 6 seconds. It can be 5 to 7 or 8. That's not the issue here, right? But very, very few seconds, and I'm going to use it. Right? What's the next step? What's the boost of energy? This is creatine phosphate, right? This is when we're going to use creatine phosphate. We'll show it in a second. But it's a wonderful, very short-term, but very efficient way to get your energy. Okay? But we are losing, again, not losing, but we are basically breaking down all the creatine phosphate, so we have just creatine now. We need to replenish, in a way, the resource. So 10 seconds. The next step, glycolysis, where I'm gonna, what's the source for glycolysis? What do I need? Glucose, where I'm gonna get? Skeletal muscles. I have glycogen for that, right? And finally, I will need, what's the difference between this part and this part? This is aerobic versus anaerobic respiration, right? You can go back to your slides. So first of all, the ATP that is stored in the muscles, then ATP with creatine phosphate or the phosphagen system. Glycogen will be converted to glucose, and then we'll have glycolysis. And whether glycolysis is just staying as a glycolysis, fermentation, and everything, we're not going to go into the biochemistry. You don't need to know the reactions. That's fine for, let's say, 40 seconds, a minute or so. You're running for a 100-meter race, right? It's going to be, I mean, if you're not using both, this will be sufficient. You'll just start to use glycolysis, okay? But then at some point, you'll need aerobic respiration. The next slides are very brief. It's just to show, again, this is something you should know at this point, creatine phosphate, that can, basically we can couple the breakdown of creatine phosphate to creatine phosphate. We can couple it and add phosphate, P, to ADP to create ATP. I will just say one word about glycolysis. Again, this is not biochemistry. You don't need to remember glycolysis and fermentation. I do want to say a word about the fact that if you don't have oxygen, right, you are going eventually to end with lactic acid, okay? And I just want to mention this because this is still yet a strong misconception, a mistake that stayed with us, unfortunately, that many people think that if you exercise a lot, then you feel the pain, which you will feel the pain. It's because of lactic acid accumulation. It is true that acid in an acidic environment does weaken the whole system, right? That's one thing, but it's not lactic acid. Lactic acid is easily removed from the muscle. It has a very short half-life time, and it's going to be converted actually in the liver back to glucose, so we're going to use it. Okay, so unless there's something abnormal, normal conditions, it's not lactic acid that makes you fatigue. Okay, and if you do have oxygen, okay, those who took biochemistry, what happens instead of lactic acid from pyruvic acid? What's the next step? Acetyl-CoA, sounds familiar? Not for the exam, not for you, not for 252, okay? And we go to the mitochondria, which is much more efficient. So the one thing I do want to add before we finish is that, okay, this is all great. We need the oxygen, but we need to get the oxygen from. So where do we get the oxygen from? We get it from the air, but where is it there for us? So first is going to be myoglobin. Before we use the hemoglobin coming from the oxygen coming from the lungs, blood and from the lungs originally, we use myoglobin. Myoglobin is kind of like a cousin, if you will, of hemoglobin. It's very similar in structure, okay? When it has the oxygenated iron, it's also red. It has a red pigment, and this is why your muscles are red, okay? And if you eat meat, and by the end of this class, hopefully none of you will actually eat meat after I'll convert you all to veganism, then those who eat meat, or just look at meat, right? On muscles, you'll see that you have red meat, right, and white meat. This is not because more blood, less blood. This is not the color. The color is how much myoglobin you have, basically. The second source, of course, is the cardiovascular and respiratory system. And you read about, and I will probably ask you about endurance versus heavyweight, what you gain. Okay, I will just say that the more exercise, you get better blood supply and better usage of the oxygen. And the last slide, I'm sorry, I'm stealing just 1 minute, okay? Just to emphasize, all motor units show a twitch pattern, but there is more than one type of twitch, and muscles will have different types of motor units. The distribution of the motor units is different. We distinguish between slow oxidative red fibers. Why? Because we need them for a longer time. So we need oxygen, and we need a lot of myoglobin, so they are red, so they are slow, but more prolonged, versus the fast glycolytic when we want something that is fast, so we don't need the whole aerobic thing, we just need glycolysis. They're going to be fast. We don't need to have a lot of myoglobin, so they will be, sometimes we call them white fibers. You don't need to know to remember the type 1, type 2 or so, but you need to be familiar with the slow oxidative and fast glycolytic. Okay? Again, have a great weekend, and we'll send you